Hi, everyone. Welcome to our seminars on the cerebellum. As you know, when we age, we have a decline in our motor function and cognitive function. And often this is accompanied with DNA damage that accumulates. A universal anti-aging intervention is dietary restriction, but how does dietary restriction produce improvements in longevity? Dick Jaarsma has been studying neurodegeneration and aging and studying the role of dietary restriction, particularly its effects on the cerebellum. He started his studies with Jacob Korf in biological psychiatry in the University of Groningen, then with Enrico Mugnani at Northwestern University, studying cholinergic intervention of the cerebellum and the physiology of unipolar brush cells, after which he returned to the Netherlands, where he is now a group leader at Erasmus Medical College in Rotterdam. It's great to have you with us, Dick. Thank you so much.
Okay, thank you for the invitation. And Martijn warned me that you are a very physiological group. Is that the case? Anyway, I will start. So is it the? Are you going to ask questions in the middle or at the end?
Whatever you like.
Okay. Now, if something is not clear, you just can ask questions. So today I will share two stories with you. One is indeed about dietary restriction, and mainly we use a progeric DNA repair deficient mouse model, and where dietary restriction has a big effect. And the second half, or at the end, I will talk a little bit about the new study, which is not published, but it's a mouse model where there's a striking amount of cerebellar pathology, mainly in the granular cell layer. It's very pathological and molecular, but okay, let's start. So it's more and more clear that if you have problems with DNA repair pathways, in many occasions you have neurodegenerative or neurodevelopmental problems. So DNA repair is important for maintaining your brain, and there is multiple pathways. I will mainly talk about one pathway, which is the NER pathway, nucleotide excision repair pathway. However, these pathways, at least components of the pathways, operate in multiple pathways. So before I start, a short remark about dietary restriction. So it's basically still the only well-established universal anti-aging intervention, and it works in many organisms, including primates, as shown here. This is a rhesus monkey. There have been two big, very long studies going on, and now the animals are reaching the age of 40, and it becomes more and more clear that also in these primates, both health span and lifespan is increased, and the oldest mouse died at I think at 43 years, which is above 120 years if you would compare it with humans. What's important to realize is that it also is very beneficial for the nervous system in these monkeys. The mechanism is not yet precisely known, although there's a lot of knowledge, but it has been hard to reproduce the effect of diet restriction with intervention that operate on pathways presumed to mediate the effect of diet restriction. So we don't have a full understanding of what's going on. And it's also, most experiments, at least in mammals, are done in mice. And in mice, you typically achieve about 30% increase in lifespan when you do severe diet restriction. So I became involved in the project when the people from the genetics department, molecular genetics department, Jan Huymaker and Wilbert Vermeij, together with the RIVM, it's a Dutch institute, were performing diet restriction studies with the ECC1 delta minus mouse model. So ECC1 is a component of the NER pathway, and but it also plays a role in other DNA repair pathways. And what is apparent from this mouse is that it stays small and it has degeneration in many systems, and including neurological decline, and it dies at about after 6 months. And in the molecular genetics department, but also some groups at the NIH in the US, they use this as an aging mouse model. And the nice thing is that it ages faster than the normal mouse. So you can, so lifespan analysis and health span analysis takes less time. So a little bit unexpected, diet restriction showed a dramatic beneficial effect. So that has been published some years ago. And it's more than doubling the lifespan. And it's more or less linear. So you can also do 2 weeks of diet restriction, then lifespan is increased by 2 weeks. You can do 4 weeks diet restriction, then lifespan is increased by 4 weeks. So in a way, it seems to stop it looks like it stops the degenerative process. And the effect is very large on neurological readouts. So when the normal mice are nearly dead, or dying, moribund, the diet restricted mice still are capable of normal walking and are relatively healthy. And you have to realize that the mice are very small, because they are both small by themselves, and then the diet restriction even make them smaller. Okay, the effect is very clear on rotor rod. This is at 60 weeks. So one of them is the ECC1 on diet restriction, and you see the ad libitum mouse is falling immediately, and then the wild type mouse is cannot cope with the rotor rod. But most ECC1 mouse on diet restriction can perform the rotor rod near as good as wild type mice. And this goes on for very long after the normal survival time of the ad libitum mouse. So it's really a very strong phenotype. And another marker we analyzed is this is the result from a transcriptomic analysis. And what was very clear, that one of the phenotypes in these mice is that there's a selective downregulation in many tissues of long genes. The idea is that because long genes are statistically more prone to errors, to have damage, and so there's an increased risk of in the absence of repair, there is an increased risk of problems. And this has now been also seen in normal aging and in other mouse models, and it's called gene length dependent transcription decline. And basically, it's an indirect readout of DNA damage that interferes with transcription. And if you look at this readout, so here you see the ad libitum animal, and you see that there is a relatively high level of long genes, so between larger than 500 kilobase that show a downregulation, whereas in the diet restriction animals, this phenotype is not present. So this has been interpreted that diet restriction indeed preserves the quality of the DNA and prevents problems with transcription. Okay, so then of course we try to find out what's going on, and then you can do all kinds of experiments with known dietary restriction mimetics that are also now more or less in the clinic, some of them. Whether they work is still not clear. But one of the best examined and most prominent in the literature anti-aging compound is rapamycin, which inhibits the mTOR pathway, and in mice has a variable beneficial effect. So we spend a lot of energy trying these compounds, but we didn't find any effect. And in some cases, there was even an adverse effect of rapamycin. So we tried different doses. We tried knockout mice of the mTOR pathway, but there was no link between the protective effect. So we could not reproduce any aspect of the protective effect of diet restriction by manipulating the mTOR pathway. So the idea is that mTOR doesn't play a role. And this also appeared from the pathological studies. So we quantified Purkinje cells, and with diet restriction, you see preservation of the Purkinje cells, but not with rapamycin. Although with rapamycin, we showed that we indeed can downregulate the mTOR pathway. So that the rapamycin has a biological effect, but it doesn't interfere with survival in this mouse model. So then we tried many other treatments, and there's only one which did a little bit, and that's stimulating the NAD metabolism by adding NAD precursors to the food, and one of them had a little bit of a beneficial effect. So this is a work in progress. We are looking in more detail at this pathway. So then the question many people ask: so how do you know that diet restriction or how do you know that degeneration in these mice in the brain is linked, is a cell autonomous effect? So because maybe there's some papers suggesting that brain pathology in these mice result secondarily from kidney pathology or liver pathology. So we started to make neuron-specific mouse models, including a Purkinje cell-specific mouse model. And then you see severe selective Purkinje cell pathology. And then when you test diet restriction, you see that you can equally well protect the neurons from degeneration. So, and this is also illustrated here. So the idea is that diet restriction via yet to be defined mechanisms targets cell intrinsic DNA damage driven Purkinje cell degeneration. So somehow it changes something in the Purkinje cell that it becomes less vulnerable to the absence of this DNA repair pathway. Okay, so then the question is, what happens in the cerebellum, what happens in Purkinje cell when you do dietary restriction? And it's important to realize that the brain behaves a little bit different compared to other tissue. The brain maintains its normal weight, and basically the brain, all the sugar, all the energy which is still present goes to the brain. You cannot just turn the brain off. It doesn't shrink. Then, in addition, there was a study, a big study on transcriptome changes triggered by dietary restriction in multiple tissues. This is an old study done with microarrays, and basically a lot of genes changed in many tissues and also in some brain areas, but there was no change found in this, no consistent change found in the cerebellum. So the idea was that there is no change in the cerebellum. There is some evidence that there is activation of inhibition of mTOR, which is well known to occur, but as I said, that could not explain our previous data suggested that could not explain the protective effect of DR in our animals, because rapamycin didn't have any effect. So just to check whether maybe there was some subtle changes, and we did a small RNA-seq analysis, which is a little bit more sensitive than the microarray studies, and we tried to do it very clean, very precise, and then indeed we found differentially expressed genes which were consistently differentially expressed between dietary restricted animals and ad libitum fed animals. And so you have differentially expressed genes, but you can also just look at gene sets and whether they behave in a certain way. And what was clear that gene sets that were linked to synaptic transmission consistently went down, whereas gene sets that were involved in maintenance and repair consistently went up. And that's also illustrated here. So this is ingenuity pathway analysis with differentially expressed genes. So you are biased. So here we only looked at differentially expressed genes, which were significantly different between groups. And then you can see different types of pathways. But what is very clear that many of these pathways are linked to signaling, calcium signaling, excitatory signaling, synaptic plasticity. And this is also true for gene set enrichment analysis. And then if you look at upregulated genes, they mostly were linked to maintenance and repair. Okay, and this fits the general idea that globally diet restriction shifts the focus of the cells from physiology to maintenance and repair. What happens during diet restriction in Purkinje cells, and whether you can measure that in the physiology or in the size and so on, we still have to find out. Okay. So we also did gene set enrichment analysis with cell-specific genes derived from single nuclear RNA-seq studies. This was the first study, and then you can they identify genes specific for Purkinje cells and genes specific for granular cells. And if you do gene set enrichment analysis, so for these, you see that there is a large bias of downregulation for Purkinje cell genes and not such a bias for granular cell genes. And the same is true for other gene sets. For instance, also the gene sets from the large Kosovo studies, they show a downregulation of Purkinje cell genes and no effect on granular cell genes. So that's where we are now with the diet restriction studies. Funding finished, PhD studies were gone, so we are now in the process of looking how to progress and how to continue these studies. So now the second part of my talk is something completely different. How much time do I have? And this is more about the question, what precisely is going on in these brain in the disorders linked to the nucleotide excision repair pathway. And the nucleotide excision repair pathway is a complex pathway with many components and many steps. And an important complexity is the division of two initial damage recognition steps. And one is called transcription-coupled nucleotide excision repair, or TC-NAIR. And that's a pathway which is linked to RNA polymerase II, which progresses and transcribes RNA, and then at a certain point encounters a transcription-blocking lesion, so it cannot progress anymore. And then a cascade of events start, beginning with the recruitment of these two proteins, CSB and CSA. And then RNA polymerase is removed from the lesion, and the lesion is prepared, and there will be an excision, and then there will be a repair of the DNA fragment. So then the other pathway is global genome repair pathway, and that's linked to proteins, to two proteins, XPC and DDB1 and 2, that together continuously screen the genome. And this pathway is not linked to transcription. The general idea is that the TC-NAIR pathway is very important for neurons. If that's damaged, you have problems with transcription. And if you have a problem with the GG-NAIR pathway, you have problems with accumulation of mutations, which then in the long term can result in cancer. So patients with mutations in this pathway have increased cancer vulnerability, especially for UV-induced cancer. The skin is very sensitive for UV because this pathway, the NAIR pathway, is very important for UV damage. So also the deficiency in TC-NAIR causes UV sensitivity, but these patients don't have cancer. They just get severe skin burn with minimal UV exposure. Cohowever, in most occasions, these patients develop a severe condition, which is called the Cockayne syndrome, and that can be caused either by CSA, which is here, or CSB, which have different functions. And the The syndrome is characterized by growth failure, accelerated aging, and neurological problems, and some other problems which are more variable. But nervous system problems are a constant hallmark, and they are progressive, and the patients die prematurely, and at the end of, depending a little bit on the form, but at the end they have severe neurological dysfunction. Yeah, another point which is becoming more and more evident is that these initial steps are not only for the TC-NAIR pathway, but they can also operate in other pathways. So they play a role whenever RNA polymerase is stalled. They are recruited, and then they can also trigger all the pathways, although this is a field which is still in progress. The key question now is, so it's very well known that this pathway is important for UV, but that cannot explain the damage in the nervous system. So one of the questions is what's going on in the nervous system, and what is the main metabolite causing damage, and are there several metabolites, and what's going on? And overall, the general hypothesis is that oxidative stress mediated damage was a key problem. So oxygen radicals, directly or indirectly, can generate different types of damage, and some of them can interfere with transcription. And the general idea was that this was a major factor in causing cocaine syndrome. Oh, yeah, sorry, this is the reverse. So then, yeah, it became clear that CSB deficient cells were sensitive for formaldehyde, and it can be directly tested by adding formaldehyde to the cells, and then you will see that CSB cells are vulnerable, but CSB cells without the detoxifying enzyme ADH5 are much more vulnerable. So formaldehyde is here. It is a metabolite which is comes from different metabolic pathways, also linked to glycine metabolism and other steps. And then it detoxified by ADH5, and which it converts it into formate. So K.J. Patel and Juan Guadalajara started to study mouse models which were deficient both in CSB and in ADH5. And that's basically the starting point for this project. So what they showed is that the mice which have a relatively mild phenotype when they are single mutants became much more severely ill and developed a cocaine syndrome-like phenotype, which was much more similar to the human phenotype than the single mutant. And that this phenotype included severe neurological problems. It's a little bit slower than the mouse I showed before, and they can still become quite old, but they are severely ataxic before 1 year of age. And the animals become small and severely abnormal. So then I started to collaborate with them because I wanted to see to what extent the pathology in the double mutant mimics the pathology in the single mutant, and whether there was something specific to be seen, and also to compare the pathology of this mouse model with the other NAIR deficient mouse models I was studying. And one aspect which was clear is that, so if you look at the cocaine syndrome mouse model, it has already a little bit higher, more pathology in the cerebellar guanidine cell layer. And what is studied here is nuclear accumulation of p53, which is a very important protein in DNA damage signaling pathways. And it accumulates following several DNA damage response pathways. But then if you look at the double mutant, there's much more p53 accumulation, and it occurs in Purkinje cells and other cells, but what is very striking, there's really a lot of cells in the guanidine cell layer, and most of them are guanidine cells. So we plotted them, we compared them with other NAIR mouse mutants. But the key finding is that there's a lot of pathology in the guanidine cell layer. And we quantified that. So here it looks like there's more pathology in most brain areas, but you have to look at the numbers. And in the guanidine cell layer, the numbers are one order of magnitude larger than in other areas and compared to other mouse models. So there's a quite robust accumulation. And so then you could say, look, the guanidine cell layer is very crowded. So if you just have random accumulation of pathology in guanidine cells, then there's a lot of more cells in that region than compared to other regions. So we compared the pathology in the guanidine cell layer of the cerebellum with another guanidine cell layer, where you have the same density of neurons, which is the dentate gyrus of the hippocampus. And if you look in the other NAIR mutants, like the mouse I just presented, so that this multi-pathway damage mouse model, which has pathology basically throughout the brain, and you have equal numbers of p53 cells, or the same order of magnitude of p53 cells in the dentate gyrus guanidine cells as compared to cerebellar guanidine cells. But that's not the case for the cocaine syndrome mouse model, the double mutant, which has much higher pathology in the guanidine cell layer, the guanidine cell layer of the cerebellum. So, okay, so there was one result. Then we moved to another mouse model, which we anticipated could interfere with the amount of oxygen-free radicals. And it's a mouse model which is deficient for SOD1, which is the major oxygen dismutase outside the mitochondria. So you have SOD3, which is in the extracellular space, SOD2, which is in mitochondria, and which is very important for mitochondrial maintenance. And then you have SOD1, which basically is in the nucleus and in the rest of the cytosol. So we predicted that also this double mutant would have more pathology, but that's not the case. So the pathology is the same as in the single mutant. So increasing the level of oxygen radicals by generating this double mutant does not increase the pathology in the cocaine syndrome mouse model. So now we are still in the process of looking at other markers. This is a marker, so there's gliosis and other pathology, but this is a pathology we do not fully understand. It's accumulation of p62, which is an important protein in organelle degradation and protein degradation, and it targets organelles and proteins for the autophagy pathway. And if you have an overload of autophagy, so if you block out the autophagic pathways, you see this kind of accumulation. So somehow, and this is really a dramatic phenotype. So it's basically, yeah, it's a dramatic phenotype. It's much more abundant than the p53 even, and it's very limited in the single mutant. But what exactly underlies this accumulation? It looks like the cell has a lot of, it's either a stress response or it starts to degrade itself, or that's something we have to find out. So basically, this is the, I'm nearly done. So the conclusions is from these two studies is that we have now a strong evidence that diet restriction has impact on DNA damage-driven neuronal degeneration. So, and it does so, so it operates on intrinsic damage rather than operating via other organs. And yeah, it might be because it turns down the physiology a little bit, or via mechanisms we still do not fully understand. So this is work in progress. Important. Another issue is that, which needs further attention, is that diet restriction may not operate the same in all neuronal cell types, and it triggers different gene expression in different neuronal cell types. And this is also known from comparison of different tissues. So if you compare different tissues, and there's very specific responses in different tissues when you do diet restriction. So it looks like there's cell and tissue specific pathways underlying the protective the protection. So then the second data set is about formaldehyde. So now we have strong evidence that formaldehyde is an important, endogenous formaldehyde may play an important role in the pathology of cocaine syndrome, of the nervous system pathology of cocaine syndrome. And it seems to preferentially or predominantly affect the cerebellum. And this, so there is some evidence. It's important to realize that there's not so many patients, and yeah, there's very little systematic pathological studies. But what is very constant, that is in many occasions, ataxia is an early phenomenon, and cerebellar degeneration is quite prominent in many of the cocaine syndrome patients. And that could be mediated by formaldehyde. So our suggestion would be then that you could do dietary interventions that maybe diet restriction-like interventions that lower the level of, they are predicted to lower the level of formaldehyde metabolites in the cerebellum, or as well as in other areas, to prevent the disease progression in these patients. Okay, that's it.
Thank you, Dick. That was very informative. If you have any questions for Dick, please go ahead and raise your hand, and I will give you a chance to ask questions. So you presented a little bit of data regarding behavioral consequences of dietary restrictions and their better ability to, the mice's ability to do the rotarod, for example. But when you do dietary restrictions, you reduce the energetic intake. Does it mean that the animals are also less active and more resting, or how does their behavior change?
Yeah, so that's a complicated issue. It depends a little bit on the protocol. But the animals are active in the night, and they sleep in the day. So we give them typically food at the beginning of the night. So when they have to become active, and these tests were done when they expect the food to arrive. So then they are hyperactive, they have full energy. But during the day, they sleep more, and they are much more. They are resting much more. So they have really a very a more pronounced day and night rhythm linked to the time of food arrival. And some mice, depends a little bit on the protocol, but some mice even go into torpor, which is they lower the temperature a little bit, which, do you know about torpor? Maybe.
Yes.
Yeah, torpor. So mice and other small animals have the ability to lower the temperature and to go in an even more restful state than sleep. And that's what happening. And that could also explain, basically, they are turned off for a couple of hours, something like that, and that could explain partially the strong effect of dietary restriction. So people are also trying to play with temperature, and yeah, there's also studies where they do dietary restriction, but they give the food, they prevent sleep. Then dietary restriction has a much less beneficial effect. But that could also be linked to the fact that basically we call it dietary restriction, it's also dietary restriction, but you include periods of fasting. So you have cycled. So there's many aspects to that, and that's also, I didn't tell that, but the key next step for RNA-seq analysis is to look at RNA expression throughout the day, and also linked to activity and so on and so forth.
Wonderful. Are there any questions for you? One of my students was mentioning that there was a recent article in the New York Times saying that it's unclear if dietary restrictions is effective in humans. What's your view on that?
Yeah, that's a discussion. Trials are being done. Metabolism of humans is a little bit more complicated, and these studies are very, very hard. I don't know the recent state of affairs, but there are still believers, but it's very hard to do. Yes, so you have really. But then there's epidemiological evidence that in areas where the food is limited, and where people live in very where people are skinny on some an Italian island, and there's some Japanese islands, so that there's limited food, and the idea is that people there become a little bit older, maybe because of the diet, but also they have also healthy diet and so on. So this is whether it's one-to-one translatable to humans. That's yeah, that's hard to say. Other questions?
Thank you so much, Nick. That was lovely.
Okay, thank you for having me. So I stop sharing. Yeah. And have a nice day.
Thank you so much. Have a wonderful day, everyone.